Today's topic is on lipoproteins and their metabolism and the disorders associated with this lipoprotein metabolism. Now, we have studied one of the lipoproteins that is known as the chylomicrons. Now, the chylomicrons are the dietary lipoproteins, as you all know. They are synthesized in the during the process of digestion and absorption of lipids. They are formed in the intestinal mucosal cells. The chylomicrons are the major carriers of the triacylglycerol and to a lesser extent cholesterol, the fat-soluble vitamins. These are the constituents of the chylomicrons with an apoprotein that is known as the FOB48. FOB48. So they are known as the chylomicrons. They contain the dietary triacylglycerol, cholesterol, phospholipids, fat-soluble vitamins, and these are further absorbed into the lymphatic system and then come into the portal blood. And these chylomicrons, as you know, it will give a turbid nature to the plasma after about 2 to 3 hours of your food intake. So if you collect the blood after about 2 to 3 hours after your food intake, you can see that the plasma is not clear, rather it is milky or turbid due to the presence of chylomicrons. Now after another 1 or 2 hours, you can see that these chylomicrons they disappear and the plasma becomes transparent. It becomes clear. That means the chylomicrons have been removed with the help of lipoprotein lipase. This lipoprotein lipase is an enzyme which is present in the capillary walls of various tissues. Lipoprotein lipase sequentially removes the fatty acids from the triacylglycerol or triglycerides, and the fatty acids are produced, and the free fatty acids they go into various tissues, and that glycerol is also separated. So this part you have studied during the digestion process. Now the chylomicrons have the least density. They have only 2% proteins, only 2% protein, and as good as 98% lipids. So the lipids are more and protein is less. So naturally, the density will be very low, and so the chylomicrons will be floating in the top layer, right? So chylomicrons have the least density. Now, the other lipoproteins, in addition to the chylomicrons, you can see that many of you may be knowing also. So one is VLDL, then you have IDL, LDL, and HDL. So first one is chylomicron. So as and when you go from chylomicrons to VLDL, IDL, LDL, and HDL, the density increases because the protein content also increases. So here the protein content is about 2%, here it is about 10%, here it is about 12%, that is this one, and for LDL it is about 16%, and HDL has got a protein percentage of 60%, and relatively the lipid content will be less. And if you consider the major lipids in kilomicrons, it is TAG, cholesterol ester, you have phospholipid, and fat soluble vitamins. That is the content. Now VLDL also has got triglycerides as the major lipid. Then it also has got cholesterol ester and free cholesterol also, cholesterol ester and free cholesterol, and it has got some phospholipid also. So both these lipoproteins, that is the the chylomicrons and the VLDL, they are the major, their major content is TAG, as you can see. Now IDL will have less TAG, and it will have relatively more cholesterol ester. Phospholipids will be the same as VLDL, but here the change what has taken place is that the triacylglycerol has reduced. It has reduced, and relatively the cholesterol ester has increased. Why the TAG has decreased? It is because of the action of lipoprotein lipase, which actually removes the fatty acids after hydrolysis. Now LDL, as you near the formation of LDL, it will be more of cholesterol, and there will be as good as no TAG left. So main carrier of cholesterol is LDL. So the major lipid content of LDL is cholesterol. So it is a cholesterol rich lipoprotein, LDL. And HDL, if you consider, there is cholesterol ester present, phospholipids are there, and there is one enzyme known as LCAT. There is one enzyme known as LCAT, that is lecithin cholesterol acid transferase. So that is the composition of the. Now, if you come to this, the lipoprotein metabolism, you can see that the VLDL, this is VLDL. VLDL, nascent VLDL. It is known as nascent VLDL. Nascent means newly formed. Nascent means newly formed. It is formed from the liver. It's formed in the liver, hepatocytes, VLDL. So the nascent VLDL, after formation in the liver, which mainly consists of triglycerides, some extent cholesterol ester, but the triglycerides are in higher concentration. The triglycerides are in higher concentration. Now, the formation of VLDL is also required to remove the fat from the liver. See, the fat is not stored in the liver. Fat is stored in the adipose tissue, and fat should not be stored in the liver. So otherwise it may cause fatty liver. So we have to prevent fatty liver. So one process is to form the VLDL and export it out, secrete it out. So liver plays a major role in the lipoprotein metabolism because it forms the VLDL Not only that, it forms the bile salts for the purpose of digestion of emulsification and digestion. So there are so many important functions of the liver. As far as lipid metabolism is concerned, this is true of carbohydrate metabolism also. Glycogenolysis, glycogenesis, gluconeogenesis. Protein metabolism also, that is a urea cycle. Many important functions are concentrated in the liver. So here, as far as lipids are concerned, it is the VLDL formation that is most important. And VLDL is formed as a nascent VLDL, and also you may call it as an immature VLDL. It contains maximum triacylglycerol or triglyceride, then cholesterol ester. Now, and it has got one apoprotein known as ApoB100. Chylomicrons had ApoB48. The mRNA, mRNA for both these apoproteins are the same. But the translation of mRNA stops midway in the small intestine, whereas it goes to the full length in the liver. So in the liver, B100 is formed, and in the small intestine, it is the ApoB48. This is also known as, mRNA editing of the mRNA. It's a way of gene regulation. Right. So originally it had only ApoB100, but again, when it passes through the plasma, it receives some other apoproteins like ApoA, ApoC2, and ApoE. It receives all these apoproteins. What are these? C2 and E. C2 and E, not ApoA. Okay, now C2 is required for the activation of lipoprotein lipase. This lipoprotein lipase, it is anchored in the capillary walls of various tissues like, for example, adipose tissue. The VLDL passes through the, through the blood to the various tissues, and then the ApoC2 returns back. ApoC2, the function is to activate the lipoprotein lipase. Once the lipoprotein lipase is activated, the C2 is not required. And so the C2 returns back. C2 returns back to HDL, so to the original place. So what all apoproteins are left now? ApoE is there, ApoB100 is there, TG, a lot of it is there, and cholesterol ester is there. Now the lipoprotein lipase acts on this VLDL. What change takes place? The free fatty acids are removed. And glycerol is also, glycerol also will be left. So free fatty acids goes into the plasma, into the tissues, and glycerol remains in the plasma. So with this process, what is produced is IDL. So with this, you get an intermediate density lipoprotein. Now here you can see that the content of cholesterol is more than that of TG. Here TG was born, here cholesterol esters are born. Because due to a sequential breakdown of the VLDL, of the triglycerides present in VLDL, the triglyceride content relatively reduces and relative increase of cholesterol ester takes place. So that is intermediate density because it has become from very low density lipoprotein, the density has actually increased because of the removal of triacylglycerol. Now in the next step, you can see that the ApoE is returning back to the HDL. FOE also returns back to the HDL. So FOC has returned and FOE also has gone back. So C and E, which VLDL had received from HDL, have gone back. Now, this IDL further undergoes the action of lipoprotein lipase, and almost complete TAG is removed. And almost complete TAG is removed. And what is now left is cholesterol ester. So you can say that LDL is a cholesterol-laden, cholesterol-rich lipoprotein, LDL, low density lipoprotein. So it has got only FOB 100, 1. So from VLDL via the ideal, LDL has been produced. So this is because of the action of lipoprotein lipase, which removes the free fatty acids from the TAG. So lipoprotein lipase is a different lipase in comparison to other lipases which you have studied, that is the lingual lipase, gastric lipase, pancreatic lipase, intestinal lipase. All of these are involved in the digestion process, digestion of lipids, whereas lipoprotein lipase is involved in the hydrolysis of VLDL and also chylomicron. So this is a different lipoprotein. Now yet another lipase is hormone sensitive lipase. Yet another lipase is hormone sensitive lipase, which is located in the adipose tissue, and it is sensitive to the function of hormones. Like glucagon will stimulate hormone sensitive lipase, whereas insulin will inhibit hormone sensitive lipase. So that's a different one. So different lipase. Now you can see that the LDL is formed. Now LDL, so from this you can make out the LDL transports the cholesterol from the liver. LDL transports cholesterol from the liver to the extrahepatic tissues. LDL transports transports the cholesterol from the liver to the extrahepatic tissues, right? Whereas HDL transports cholesterol from the peripheral tissues to the liver, right? So you can see that now this LDL will be taken up by various receptors into the extrahepatic tissues. Now for that you have to look here. This is the uptake of LDL, right? Now it is taken up by Ldl receptors. The LDL has got specific receptors known as Ldl receptors. They are located in clathrin coated pits. They are located, clathrin is a protein, so it is located in the clathrin coated pits. So the green ones are the receptors, which is present in the. So LDL combines with the Ldl receptor to form an LDL plus Ldl receptor complex. It is endocytosed, and it forms a vesicle. Then this vesicle, is known as the endosome, and from this the receptors are released and it goes back to the cell membrane to be located again in the clathrin coated pits, so that it can bring more LDL inside. So LDL contains what now? LDL contains mainly cholesterol ester, some fatty acid, and a protein is there. One protein is also there. So that protein is the apoprotein. That apoprotein B100. So that apoprotein is converted to amino acids because it fuses with lysosomes. Now the endosome fuses with the lysosomes. Now lysosomes are cellular particles which contains all the hydrolytic enzymes. It contains proteases, it contains lipases, phospholipases. All the hydrolytic enzymes are present in the lysosomes. So the lysosomal enzymes, especially the proteases, act on the apoprotein B100 to hydrolyze it to the, when proteins are hydrolyzed, what do you get? Amino acids. So amino acids are produced. A bit of triglyceride may be remaining, triglyceride also. So that will be hydrolyzed to fatty acids. And then, it will be converted, it will be the major portion will be the cholesterol. Now cholesterol, it can be either re-esterified, and stored in the, for future use. So what are the future uses of cholesterol? Future uses of cholesterol include to form the cell membrane. It may form the vitamin D. It may form the bile salts and steroid hormones. Various types of steroid hormones it can be produced. So, or it may be esterified with a fatty acid and stored if there is no immediate use. Now, this cholesterol, which is now absorbed, it can inhibit the synthesis of cholesterol, endogenous synthesis of cholesterol from acetyl-CoA. Acetyl-CoA is the precursor of cholesterol synthesis. Acetyl-CoA, through a series of steps, almost 18 or 19 steps, it will form acetoacetyl-CoA, then HMG-CoA, then HMG-CoA reductase. Then it will further go down and forms a cholesterol which contains 27 carbons. Acetyl-CoA has got 2 carbons and cholesterol has got 27. It's a steroid. The steroidal type of molecule, that is the cholesterol. So, but then if already sufficient cholesterol is present, there is no need of further synthesis. So cholesterol presence inhibits the HMG-CoA reductase. The HMG-CoA reductase enzyme, it will inhibit the HMG-CoA reductase. In ketogenesis also you have heard about HMG-CoA, where it is acted upon by HMG-CoA lyase. So the rate limiting enzyme of cholesterol biosynthesis is HMG-CoA reductase. The statin group of drugs also act on HMG-CoA reductase so that it will be inhibited and then very little cholesterol will be synthesized. Because there is no need of cholesterol now, it is already present. An extra amount of cholesterol synthesis will only worsen the situation. Because the cholesterol may be deposited and xanthoma can be caused in various tissues. Okay, so that is one way of regulation of cholesterol synthesis. Now, there are Ldl receptors. These Ldl receptors present in the clathrin-coated pits. Their synthesis also can be controlled. So one, endogenous synthesis of cholesterol is controlled. So no more extra cholesterol is formed. Again, you can reduce the number of receptors. Suppose a cell has got 1000 receptors. So it can be down regulated. It can be down regulated to maybe 500 or 200 or 100. So that should take place at the gene level. So LDL receptor gene is that. LDL receptors are proteins. They are proteins. So every protein has got a gene, a corresponding gene. So if that gene is transcribed, you get a mRNA of that gene. How the information flows? The gene information flows to mRNA, and then the mRNA will be translated into protein. That is the LDL receptor protein. So it's a protein. So our body has got about 30,000 proteins in our body. So there should be a minimum of 30,000 genes also. One gene, one protein, oblique one polypeptide. Because some genes, some proteins contain more number of polypeptides. For example, hemoglobin has got two alpha chain and beta chain. Immunoglobulins have light and heavy chains. Creatine kinase has M polypeptide and B. LDH has got 4 polypeptides. So, one gene, one polypeptide. Hypothesis. So, you should have that many number of genes as many proteins you have. So, you may have around 30,000 proteins. So, 30,000 genes have to be there. Minimum. Okay, so the cholesterol that is formed will act like a steroid hormone. Cholesterol is like a steroid. So it will bind with cytoplasmic receptors, and then it will go to the DNA and will bind with specific regions on the DNA known as HRE, hormone response elements. It will bind to specific regions on the gene known as hormone responsive elements, or HRE. So binding of this cholesterol to the HRE will lead to the formation of mRNA and then the protein. And the protein will be formed. So in this manner also at the genetic level, the cholesterol synthesis can be, cholesterol uptake, not synthesis, uptake can be regulated. How? By regulating what it will regulate? LDL receptors. So if more LDL receptors are present, so more synthesis of it will be stopped. The synthesis of new LDL receptors will be stopped. Suppose there is not much cholesterol within the cell. So there is no inhibition. It will not bind with the cytoplasmic receptors, and this will not bind to the hormone response elements. So the hormone response elements will undertake the transcription process. What is transcription? Transcription is the formation of messenger RNA from the DNA, from the gene. So from the gene, if messenger RNA is not formed, then protein also will not be formed, because you need mRNA for the synthesis of proteins, right? This part you know. So the control of cholesterol that is present in the cell is two ways. One is it will inhibit the HMG-CoA reductase step so that less cholesterol is formed, and the uptake of new cholesterol into the cell is prevented. That is by reduction of the number of LDL receptors. So it's a two-way control. It's a two-way control. So now this is a very regulated uptake. This uptake is a very regulated uptake through the clathrin-coated pits. Right? Now, sometimes this LDL may be modified. LDL can be modified. It may become oxidized LDL. The LDL may become oxidized or glycated. Right? Now, oxidized LDL can be formed when there is more presence of free radicals. Free radicals. That means one free radical is superoxide anion. Is a free radical. Lipid peroxide, hydrogen peroxide. Free radicals are being produced during the metabolic reactions. So to prevent their action, what do we have? Glutathione, that is the antioxidant. So we have antioxidants. Antioxidant vitamins are there. Beta carotene, vitamin E, and vitamin C. So they are all antioxidants. And also we have antioxidant substances like glutathione, which will convert H2O2 to water. H2O2 is harmful. H2O is not harmful. So that means there should be a balance between free radicals and antioxidants. So if that balance is tilted and antioxidants are less and more of free radicals are present. What is free radical? It has got an unpaired electron in the outer orbit. So that makes it very active. So it may destroy the biomolecules, it may destroy cell membrane, it may destroy phospholipids, it may destroy DNA. So free radicals are considered to be a cause of cancer also. This may lead to cancer or maybe diabetes mellitus, or many of the diseases, the basis is now found to be the excess of free radicals. So to counteract the free radicals, we should have enough antioxidants. If that balance is not taking place, then it becomes, a disease may be caused, or cataract also can be caused. Atherosclerosis can be caused by free radicals. So, when there are more number of free radicals, the protein may get, lipid part may get oxidized, so it forms oxidized LDL. And glycated LDL can be there when glycated LDL in hyperglycemia. When there is hyperglycemia, diabetes mellitus, the glucose that is present in excess can join with the LDL, apoprotein, and get glycated, like glycated hemoglobin. So there will be more excess of glycated LDL and oxidized LDL. Now these two are abnormal LDL. These two are abnormal LDL, and so they are not absorbed in this manner. This oxidized LDL and glycated LDL does not follow this mechanism. This mechanism is a controlled one, regulated one. Whereas, and there is a feedback also, feedback control. Whereas here, if this happens, there will be an uncontrolled uptake, unregulated uptake of LDL. Unregulated, and that is done by special cells macrophages. So these macrophages, they take up large amounts of LDL cholesterol, especially the oxidized and glycated, right? Unoxidized and non-glycated LDL are taken up by regulated mechanism. But when once it becomes oxidized or glycated, then the macrophages has a role to take up more and more LDL cholesterol, and these macrophages, after absorbing more and more cholesterol, will become form cells. FOM, form cells. So form cells, what do they contain? They contain large amounts of cholesterol. And these form cells then further migrate into the subintimal layer of the arteries, and they start getting deposited in the arterial walls. These form cells, which contains maximum cholesterol, they start getting deposited in the arterial walls of the arteries. So what is this? What this will lead to? Atherosclerosis. And there will be further, cellular proliferation taking place. Collagen synthesis is stimulated, and this forms what is known as a cholesterol plaque. It will form an atherosclerotic plaque. So the deposition of this cholesterol plaque increases, further narrowing the arterial lumen, and a stage will come when there is a full block, and this can lead to the lack of blood supply to various organs. It may lead to a lack of blood supply to the heart, and it can precipitate a heart attack known as acute myocardial infarct. And it may also cause cerebral stroke. It may also cause cerebral stroke. It may cause cataract, macroangiopathy and microangiopathy. Both can be the macroangiopathy, which may lead to AMI, and microangiopathy may lead to neuropathy, peripheral neuropathy. So there are so many complications because of atherosclerosis. So two ways of uptake of LDL. One, regulated way. It is very well controlled. It does not cause much harm. Whereas the unregulated one, through the macrophages, which uptake the oxidized LDL and glycated. So that's why the diabetic patients, they are more susceptible to develop atherosclerosis. Diabetic patients are more susceptible to atherosclerosis and cardiovascular diseases. So this one is the explanation. So LDL seems to be the real culprit because it takes the cholesterol from the liver to the peripheral tissues. LDL in excess. LDL in excess. Ideally, the LDL should be below 130 milligrams. Ideally, the LDL level should be below 130 milligram in the plasma. So if it increases, then the problem. Otherwise, there is no problem. But now we have a lipoprotein which will take up the cholesterol in the reverse direction. In the reverse. That is, from the peripheral tissue back to the liver. And from the liver, it is easy to excrete the excess amount of cholesterol. From the liver, the cholesterol can be excreted through what? Through bile. So that is a way of removing the cholesterol, and also it can be converted to bile salt. So bile salt also is excreted in the bile. So that is a way of removing cholesterol. So HDL, what it does, HDL can collect the excess amount of cholesterol from peripheral tissues, and as it is formed, it is again in discoidal shape. Discoidal shape. So there is a nascent HDL. Nascent HDL. Nascent HDL, contains more of free cholesterol. Nascent HDL contains more of free cholesterol, not cholesterol ester. It contains phospholipids. It contains apoprotein A. It contains apoprotein A. What else? It contains apoprotein C, C2, and also apoprotein E. Apoprotein E. So A, C2, and E. It contains more of free cholesterol. Phospholipids are on the periphery because they are water soluble. Phospholipids are water soluble, isn't it? Phosphatidylcholine. Phosphatidylcholine is a phospholipid. That's a lecithin. Lecithin. Okay, so this HDL, they will go on collecting the cholesterol from the peripheral tissues, and it is known as scavenging action. Scavenger. So it will remove the cholesterol. So it is mostly free cholesterol, and it has to be esterified now. It has to be esterified. So you have to add a fatty acid into that. So it becomes an ester cholesterol. So where from the fatty acid comes, it is from the lecithin, this phospholipid. So free cholesterol plus lecithin in presence of LCAT enzyme, lecithin cholesterol acid transferase, it forms the free cholesterol will form cholesterol ester, and it will form another product is lyso lecithin. So now what has happened? This HDL contains more of cholesterol ester. So the HDL first formed goes on taking cholesterol, then it with the help of this enzyme, free cholesterol is and lecithin helps in the formation of cholesterol ester. So the first formed HDL is known as HDL3. First formed cholesterol is known as HDL3. It goes on collecting cholesterol and esterifies it, and it is now converted to HDL2. And this HDL2, which carries all the cholesterol which is in the ester form, goes to the liver, and in the liver it is taken out. So the HDL2 takes into the liver, and now from the liver, the excess cholesterol can be excreted. Right? So now I will ask you a few questions. So lipoprotein lipase. Who will answer? You? Or you can also answer if you come. Lipoprotein lipase.
The lipoprotein lipase, which is present in the capillary walls of muscles, it acts on the triacylglycerols, which is present in maybe the chylomicrons or low density lipoproteins, and it breaks down the triacylglycerols into free fatty acids and glycerols, further converting chylomicrons into its lower forms, or in the very low density lipoproteins into intermediate density lipoproteins, and further into low density
Okay, fine.
lipoproteins.
Okay. Now, about the mechanism of uptake of LDL, the regulated way, the clathrin coated cakes. Who will answer? Anyone? Ready? You can?
LDL, it is first of all taken up by the Ldl receptor that are present on the capillary wall, means membrane. Then it contains the clathrin peaks, so they are located over there. Then the LDL gets attached to it, and it is endocytosed. Then it will move inside, and then it will be acted, means it will be in the form of endosomes, and it will be acted upon by the lysosomes, and it will convert it into different forms. It is containing ApoB48, means it contains an apoprotein which is acted upon by the lysosomes, and so it will be converted into the amino acids, and the remaining TAG will be converted into the fatty acids.
Yeah. Okay, right. So, what is another mode of uptake other than this regulated way of uptake? I mean the macrophages. Uptake by macrophages. So, macrophage uptake, which is which requires this oxidized or glycated. Who will answer? Or anything else related to this, if you wish to answer, or if you wish to say, you can say. You can? Something? Some contribution?
Normal LDL such as glycated or with the free radicals acted LDL, that is abnormal LDL,
Oxidized
LDL, that is. They are taken by the macrophages, and they get deposited over the walls of the capillaries, arteries, and they form a plaque-like particle with the
a lot of cholesterol.
It accumulates all the particles around the capillaries and all the wall lining of the wall, and it forms it does not allow the flow of blood to the heart and various important organs. So it causes a major disease, that is atherosclerosis, which further has various complications such as cerebral stroke and all other complications.
Yeah. Okay, good, nice. What about you? You would like to?
Sir, I would like to share something about lipoprotein A.
Yeah.
Lipoprotein A is a form of LDL, and it is an analog to plasminogen. Plasminogen converts inactive plasmin to active plasmin, which degrades fibrin clots. So, as this lipoprotein A is analogous to plasminogen, so plasmin will not be activated, and so fibrin clots cannot be dissolved. So that way can also it cause atherosclerosis. So clots cannot be
dissolved.
So
Lipoprotein A is a
atherogenic
lipoprotein. Okay, nice. Good. Who else can speak something? Anybody can. You don't want to speak? can talk about the lipoprotein composition, the different type of lipoproteins.
The entire mechanism of the lipoprotein metabolism is to prevent the fat deposition in the liver that causes fatty liver and liver cirrhosis.
Yeah.
Ultimately. So what happens is the VLDL is a carrier there. So their triglycerides are more than cholesterol ester in VLDL. So what happens is after getting acted upon by the lipoprotein lipase, the triglyceride content of the VLDL it gets reduced, and cholesterol is and as a result cholesterol ester content will be more. So that form is the intermediate lipoprotein, intermediate density lipoprotein, and in that form the cholesterol ester content will be more than the triglyceride content. So and it again gets acted upon by the lipoprotein lipase, and the LDL and the resultant form is the LDL, and the cholesterol ester content in the LDL will be more, and there's a less amount of the very less amount of the triglyceride, because in the end it all gets by the lipoprotein lipase action, free fatty acids are generated which go into the adipose tissue, and the glycerol part is there. So the LDL, which takes the cholesterol from liver to the extrahepatic tissues, and the
Good, Adam. Nice. Okay, anyone else would like to contribute? So, some final comments from you? Anyone can answer. I am giving chance to all. Anyone? So, final comments.
It is very important, metabolism process of the lipoproteins are very important for prevention of different diseases which can ultimately affect the heart. So like LDL, which plays a very, it is a main culprit, as you say, that if its increased concentration of LDL can lead to different damages, like it causes the cholesterol plaque formation.
Okay, what about the role of HDL?
HDL, it helps in taking the cholesterol from the peripheral tissues to the liver, and thus it plays an important role. Means both LDL and HDL, they are having opposite roles, and thus it is also a very important.
So, if whether HDL is, why it is known as good lipoprotein?
Because it acts as a scavenger.
Correct. And removes the
Cholesterol.
and does not allow the deposition of cholesterol to form the atherosclerosis. So we need both of them, LDL in normal quantities, and also HDL is important. So a balance between these two are very important. So nice, you have listened well, and you could answer also some of the important points. Nice, good.